All right, good morning. We are headed into the second part of Unit 4 with meiosis. Double check that I've got everything on, and I'll start sharing my screen. Oh, there we go. I'm just double checking everything because a few minutes ago I think I recorded the meiosis video without any audio on it. The technology challenge is real here. I'll probably have to re-record that for y'all. But that's okay. So we're headed into the second part of Unit 4, just after meiosis. If you have not watched the Amoeba Sisters video, Genes and Alleles, recently, I highly suggest pausing it and taking a look at that. That will give you a great insight as to where we're headed, plus hopefully a smile for the morning. After you watch that Amoeba Sisters video, know that we're talking about how information is transmitted from one generation to the next. So that's where we use the term hereditary or heredity, and how we get generations that have similar characteristics, whether we're talking about something as simple as looking alike, or whether we're talking about transmission of disease from one generation to the next. And then genetics is looking at the study of that transmission. Gregor Mendel was a monk in the Catholic Church, and he was the first person to actually track a lot of these traits from one generation to the next. And he did that with looking at pea plants. It was really easy for him to manipulate these pea plants and to have quick new generations. So if we think about the generation time for humans, we're talking about the gestation period of pregnancy, which is 9 months. And usually most human pregnancies result in one offspring, one child. But that's not true for all species. Some species reproduce much quicker and have much larger number of offsprings. So a lot of genetic research is done in organisms that kind of fit that bill. So what he was able to do, he took a parent flower that was purple, and he physically crossed that purple flower with a flower that was white. And what he found was that all of the resulting offspring were purple. So the next examples are going to walk us through, well, what can we determine from that, and how do we know what genetics are at play? So when we're looking at these different traits, we call them characteristics, and we can also call them phenotypes. So with the phenotype, we're looking at outward characteristics of what the genetics are coding for. So purple flowers compared to white flowers. Other traits that Mendel looked at were if the pea pod was really full or if it was wrinkled. If the peas themselves were green or if they were yellow, and also if the peas were smooth or if they were wrinkled. You could also think about dogs in this example with different coat colors. We also know in farming that different characteristics or phenotypes are more desirable, whether it's just a larger body size or other certain characteristics of the animals. Eye color is another good example, but you can see here we have a lot of continuous variation, and this is for a number of reasons. Number one, there's multiple genes that contribute to eye color, and then those multiple genes are coding for the protein melanin, and the variation is a sliding scale of how much melanin is produced for the eye color. So it's not just all the melanin or none, there's intermediate amounts as well. So with the flower colors, true breeding means that if we took the purple flower here and had it breed with itself, it would only produce purple flowers. If we took this white flower and had it breed by itself, it would only produce white flowers. And so that means that phenotypically, those parents are going to result in breeding of the same thing. What we know is that if we cross those true breeding parents, we get all purple flowers. And so what I would like you to do is to pause the video and predict why that is. You can use terms such as dominant and recessive, and we can also start thinking about assigning an uppercase letter for dominant and a lowercase letter for a recessive trait. So go ahead and pause that video, and for the P generation and the F1 generation, try to apply those terms. Now that you've had a chance to apply those terms for the P generation and the F1 generation, we could say that the purple flowers are dominant. So the purple color is dominant compared to the white flower that's recessive. And that's because we're seeing the purple dominant trait show up in the F1 generation. And the white flower is not lost, it's just masked or covered up. And we can see the F2 generation having that white flower re-emerge. In terms of letters, in terms of the genotype to determine these, the purple flower up here you could designate as big P, and the white flower little p. I think one of the next slides will show that a little bit more clearly. And what that means is as long as you have one uppercase letter, the dominant trait will show up if we're talking about true Mendelian genetics. Something to also point out here is the F2 generation. The purple flowers have a 75% production rate, and the white flowers have show up at a 25% rate. You could also say that that's a 3-to-1 ratio. Those all mean the same thing. So what I was talking about with the letters is that these traits, these phenotypes, are determined by genes. And those genes have alleles on them. And what that means is that the allele is a different form of that specific trait. So for example, if we're back to our flower color example, that those instructions are found in the same place on both the gene from the mom and the gene from the dad. But we know that the parents can exhibit different traits, different phenotypes themselves. So the allele is a form of that specific gene, and we mentioned before that uppercase designates the dominant trait, lowercase designates the recessive trait. And so we can use another set of terms to describe, well, for a given gene, for a given letter, we can have one allele that's uppercase and another allele that's lowercase. And this means that that individual would be heterozygous. They would still show the dominant trait because they have at least one uppercase allele, but that recessive trait, the white color, for example, is still there. You could also have a situation where you have two uppercase letters And in that situation, you would see the dominant trait. We're only seeing the recessive trait, the white flower color in the flower example, if we have two lowercase alleles, so two little b's, for example. The letter that you use doesn't necessarily matter. I would just pick a letter that you can distinguish uppercase and lowercase from. All right, so back to our purple and white flowers. So what it's asking, if an individual has two different alleles, so an uppercase and a lowercase, what is it called? And you can pause the video and refer back to the previous slide. It's going to be called heterozygous because it has two different alleles. If we're using the letter P to determine flower color, and we know that a purple flower is heterozygous, it's going to be uppercase P, lowercase p. When we're writing the genotype for any given gene, we're always writing two alleles. So we would say one gene has two alleles, and those two alleles could either be the same, two uppercase alleles giving us the purple flower, or we would have two lowercase alleles, two little p's, giving us the white flower. And so we can say that the white flower is the recessive trait and the purple flower is the dominant trait. So reiterating that the phenotype is the physical characteristic that we see, freckles or no freckles, chocolate coat or black coat, purple flower or white flower. The genotype is the combination of the letters. So freckles is a dominant trait, and so for freckles to occur, big F can be the genotype. It could also be big F little f because that little f is masked in the heterozygous individual. For the chocolate coat, that is a recessive trait, so only if we see the two lowercase alleles will we get that chocolate coat. And then for the purple flower color, we could have big P, or we could have little p, either way. All right, so now it is your turn to look at how a monohybrid cross is going to work. And this monohybrid cross is predicting offspring. So back to our example of the pure breeding purple flower and pure breeding white flower, you'll want to have that picture open in your textbook. And so we're determining, okay, in that parent generation, predicting what the white flower genotype is. So go ahead and pause and determine what the genotype of that white flower is. Now that you've had a chance to determine that, we're looking at little p. I wish I had an option to kind of draw on my screen, but I think I'm limited right now. So we have big P for the purple, little p for the white, and that's going to be the genotype. There we go. So in this next cross, we're looking at, well, what if the purple flower was the heterozygous individual and we're crossing it with a white flower? That's going to give us a different result than if we had two true breeding flowers. Some other examples to clarify the different traits and how they relate to dominant and recessive. The letters that you see here are going to be your genotype, and the colors or the trait is going to be your phenotype. All right, so lastly for this section, we're going to walk through how to do Punnett squares. Majority of This chapter will be you working a lot of problems. So I'm going to do one example with the monohybrid cross, and my hope is then you can apply it to the different examples that I give you in the activities. The Amoeba Sisters also have a great video on monohybrid crosses, so I highly suggest watching that as well. So when we're looking at doing Punnett squares, on the outside are going to be the parents. So what we see here is one parent, the female, is big B, big B. And so making sure that you're making notes of what the parent genotype is and what the parent phenotype is, I think really helps this understanding. So this mom would have a black coat. She is homozygous dominant. We put one letter here and we put the other letter here because in meiosis, we are splitting those two alleles. So think about the outside of these squares as the result of meiosis. The dad, on the other hand, up here at the top is little b, little b. And that means that the dad is going to be the chocolate lab. So the lowercase b is chocolate, and we have to separate the two. Now, when we're determining the Punnett square for the resulting offspring, you're going to use this as kind of a grid. So you're going to take the little b from the dad and put it in the box, the big B from the mom and put it in the box. The capital letter will always go first, and that's a formatting thing. So within any given letter set, you'll always write the capital B first and the little b second. As you can see with this cross, we get all black labs and they're all heterozygous. This is very similar to the example with the flowers, with the purple and the white flowers. You can definitely relate it back to that. And if you want to do a Punnett square with the purple and white flowers and draw it out and compare it to this one, I think that would be a great exercise for you. So when we're talking about meiosis, what I was saying earlier is that those two alleles separate during meiosis. So this is the entire purpose of meiosis, to take the two copies of the gene and to separate them. So this is really mimicking meiosis here. One gamete will get one allele and the other gamete will get the other allele. And that's why we separate them on the outside of the Punnett square. So our problem here is to determine, well, what happens if we have a heterozygous black lab with a chocolate lab. So go ahead and pause the video and write above the slide or in your notes, what is the phenotype of these two animals, and also what is the genotype based on the information that you have. So that's step number one. Now that we've had a chance to determine the genotype and the phenotype of these two parents, the phenotype is going to be a black coat, and the phenotype for the second parent is a chocolate coat. The genotype is going to be big B, little b, if that's the letter we're choosing to use, and little b, if that's the letter we're using for chocolate. Our goal here is then to predict what the offspring will look like, and that's really what it's asking with the F1 generation. And we're going to separate that into phenotype and also genotype. Phenotype, what are the colors, and genotype, what do the letters look like? All right, so walking through this, and then I would try to do it on your own to make sure that you got the same results. So for the chocolate lab, we have one little b, and we have a second little b. For the black lab, we have an uppercase B and we have a lowercase b. If we put them together in the middle, so this little b comes in and this big B comes in, we get a black lab. Same thing over here. little b, big B. And then in these two boxes, we get little b, and the same over here. So we've got 4 squares that we can use to predict our results. So for the phenotypic ratio, you could say that, well, I've got 50% chance of having a black puppy and 50% chance of having a chocolate puppy. You could also say that that's a 1-to-1 ratio. We know, though, that these individuals have a specific genotype, so a letter combination. So in this particular example, it's the same as the phenotype, but that's not always the case. So we would say we have 50% big B, little b, 50% little b. And you can see the results down here. One of the additional parts of this is that some breeders may want to know what the genotype of their parents are, so that they can make the most money or create the best offspring. So for example, if we're talking about dog breeders, certain coat colors may be more desirable and bring more money. If we're talking about farming, then they want to make sure that they have the best parent combinations to give them the best offspring. So we can do that by performing a test cross. So say we have a phenotypically dominant animal, so we know they at least have one uppercase letter, but we don't know what that second letter is. We can do a test cross. And what that means is we're going to mate the dominant phenotype individual with a recessive individual. And so what I'd like you to do is work the cross to determine, well, if this individual was big B, and we're crossing with little b, then what would the result be? Additionally, do a second Punnett square that shows, well, what would the results be if you had big B, and we're crossing it with little b. And that's how you know what this individual is. So go ahead and pause the video and work through those 2 Punnett squares. Now that you've had a chance to work through the 2 Punnett squares, you can compare your results. So if the dominant black lab was homozygous dominant, you would get all black puppies. If the dominant lab or the black coat color was heterozygous, big B, little b, then you would get some brown puppies. Knowing that dogs have a little bit bigger litters than we do, any time that you would have even one chocolate lab would indicate that this parent would have to be big B, little b. So you've got a few different examples to go through. You've got the flower example, you've got some of the regular lab coat examples, and then you have the test cross that we went through today. I know that's a lot of information, and we usually do a lot of hands-on activities with me walking around and you all working in groups on these problems. So I highly encourage you to watch this video, watch the Amoeba Sisters, and start working on the genetics problems activities. If you have questions on this PowerPoint, send me an email and we can set up a time for you and I individually to walk through some of this. Thanks so much.